Hi, everyone. Welcome. Let me get my slides pulled up. Loading. Okay, great. I hope you all are doing well today. We have a slideshow. And if you all can drop in the chat a number 1, if you can see my slides. See what we got. Thanks, everyone. I appreciate it. All right, let's get started. I'm going to turn my camera off just to not be distracted by my own face, but we're going to have a great time today. There we go. All right, we're going to have a wonderful time today. Thank you for joining me. As Angelica said, I'm Dr. Sarah Ellis, and today, for our third installment of our Gut Microbiome Foundations webinar series, we're going to talk about the systemic effect of gut dysbiosis, kind of going beyond the belly, and talking about the different ways that the dysbiosis in the gut can impact other organs. Let me make sure I got my arrows going. Perfect. So we're going to start with the general overview of gut dysbiosis. We'll talk about the difference between a healthy and a leaky gut, and then we'll get into the fun part, which is talking about the impact of gut dysbiosis on our brain, heart, lungs, endocrine system, immune system, and genitourinary system. So we have quite a jam-packed session today, so let's jump right on in. So as an overview of gut dysbiosis, essentially, it is defined as a reduction in microbial diversity, and it features a combination of both the loss of beneficial bacteria and the rise of pathobionts. So when we look at this picture here in the green, homeostasis, you see that there are generally, equal amounts of each color or type of bacteria. It's a lot of diversity, and everything is kind of coexisting really well together. And then in dysbiosis can look like a couple of things. So on the left, dysbiosis can be caused by pathobiont expansion. So you can see the red group of bacteria has pretty much taken over about half of this area, and so we've lost a lot of other diversity, and now the pathogens are kind of beginning to take over the space. Dysbiosis can look like reduced diversity. So even though there might be a good number of bacteria, there are not a lot of different types. We see only 2 different types of bacteria here. And then here we see loss of beneficial microbes. That's another way that dysbiosis can manifest. So not necessarily the overgrowth of pathogens, those are remaining in a healthy range, but we are experiencing the loss of beneficial microbes like lactobacillus and bifidobacterium. Now, you can use a stool test to identify dysbiosis and look for dysbiosis patterns. When you run a stool test, much like our stool test called BiomeFX, you may notice a decreased diversity, both alpha and beta diversity, so a reduction in the number of bacteria, but also a reduction in the quality of bacteria as well. We'll see reduced keystone species, those special groups of bacteria that have a unique impact on our microbiome. We'll have fewer short-chain fatty acid production, and then more pathogens, more harmful metabolites and byproducts, and definitely increased inflammation. So let's talk a bit about what causes these dysbiosis risk factors. What are some things that our patients are experiencing to really cause gut dysbiosis? So here's a couple of things that we usually see. So prescription antibiotics, even natural antimicrobials, pesticides, infections, excessive alcohol intake, smoking, stress, lack of sleep, intense exercise, and even diet, especially the standard American diet or one that is rich in saturated fat. So let's take a look at our favorite schematic here at MBL. This is what a healthy gut looks like, and then we can talk about some things that happen and change in the microbiome when it becomes leaky. So in a healthy microbiome here on the left, we see that it has low levels of pathogens, a strong mucosal barrier, low mucosal inflammation, strong tight junctions. And then when we go over to see a leaky gut, we see that a leaky gut is marked by a decrease in beneficial bacteria, an overabundance of pathogens that can eat away at this mucosal layer, and particularly an overabundance of pathogens that make toxins like lipopolysaccharide. When lipopolysaccharide and these pathogens are present, they begin to wear away at this mucosal lining. It leads to weakened tight junctions, inflamed intestinal cells, and of course, the activation of the inflammatory cascade with a increase in inflammatory cytokines like IL-6, TNF alpha, and IL-1 beta. So again, when we're looking at a stool test, we can see that dysbiosis can alter the core microbiome. So we will generally see a reduction in alpha diversity, as we see here. So this patient ended up only having 98 different species of bacteria. And our goal is really to be closer to 200. So we have quite a bit of work to increase the number of bacteria in their gut. But then we also see a change in their diversity in the composition of their microbiome. So here we're seeing that she has mostly Bacteroidetes, but only a little bit of Firmicutes and quite a bit of inflammatory bacteria called proteobacteria. And when you see elevated proteobacteria, that means that's quite a bit of LPS present, and definitely a sign of dysbiosis as well. Now, dysbiosis is going to change the bacterial contribution. So the bacterial contributions of a healthy gut is very different from the contributions from a dysbiotic gut. So when we have a gut microbiome that has mostly commensals present, then we'll see short-chain fatty acids being produced like butyrate, acetate, and propionate, lots of vitamins, particularly B vitamins that are produced, antioxidants, neurotransmitters, and the gut pH is often optimized. But when a gut is experiencing dysbiosis and an overgrowth of opportunistic and pathogenic bacteria, we will see that bacteria begin to produce more harmful metabolites like ammonia, hydrogen sulfide, methane, toxins, and virulence factors that can worsen an infection, and of course, lipopolysaccharide. And we'll talk a little bit about LPS, or quite a bit about LPS, in a couple of slides. So here's what we know. We know that there are a number of conditions that have been directly correlated with gut dysbiosis. If we take a look at this picture here, we see gut dysbiosis has been directly connected with brain conditions like Parkinson's and Alzheimer's, anxiety and depression. It impacts our thyroid, particularly hypothyroidism. Musculoskeletal issues have been linked to dysbiosis. Of course, we know that gut dysbiosis definitely plays a role in certain GI conditions, but of course, metabolic disease like obesity and type 2 diabetes, and heart disease as well. When we take a look at the impact of dysbiosis of the human microbiota, we can see that there are not many areas of the body that is not impacted by gut dysbiosis. So now that we've had a really good foundation on gut dysbiosis, how it happens and what it can look like, now let's take a moment to talk about its impact on the individual organ systems that we mentioned earlier. So let's talk about dysbiosis and the gut-brain axis. All right. Our gut is powered by its own nervous system, the enteric nervous system. And there are a lot of neurons in the ENS. In fact, there are more neurons in the gut, over 100 million, than there are in the entire spinal cord. In fact, the ENS is the main neurological system that the gut is exposed to, and it is fully accessible to the microbiome and is able to operate independently of the central nervous system and peripheral nervous system as well. One of the main parts of the gut brain axis is the vagus nerve. So the gut brain axis is a bidirectional communication system between the gut and the brain. It helps regulate GI homeostasis, is able to connect emotional and cognitive areas of the brain with different gut functions. The gut brain axis includes the vagus nerve, the brain, and the spinal cord. It also includes other organ systems like the endocrine system, immune system, and of course the gut microbiome. Now, as I mentioned, the ENS, the enteric nervous system, has way more nerves than the spine, but it also produces more than 30 different neurotransmitters. And hormones and peptides that are released by the ENS can go into the blood and cross the blood-brain barrier. That's how ghrelin is able to activate our hunger hormone and activate our hunger signals and acts with the vagus nerve to facilitate proper digestion. Now, what's interesting is this gut-brain axis features both afferent and efferent signals. So think of the gut and the brain as like a friendship between an introvert and an extrovert. So what we found is that the gut to brain signals make up 90% of the signals that go between the gut and the brain. So that means that the gut is really our extroverted friend. I'm sure we all have, or we might be, that extroverted friend who always has something to share with their friends, always calling you up, telling you, what they're up to, all these amazing things that's going on in their life. And the gut is the same way. The gut is always communicating with the brain. Hey, we had this for breakfast. We had that for lunch. Oh, we might have picked up a bug and we don't feel all that well. It's constantly communicating with the brain. And the brain, on the other hand, is the introverted friend. It's the one that's nodding and saying a couple of words here or there. But when it does communicate, they say, we know with our introverted friends, they may not say much, but when they do communicate, it is something of substance and importance. And the brain is the same way. Though it may not communicate as much with the gut, when it does send a signal, it is definitely something that is of importance. So the vagus nerve is our 10th cranial nerve. It is the fastest and most direct route that connects the gut and the brain. It is also our longest nerve and it innervates quite a few different organs in our body. So it innervates the pharynx and the larynx in the neck and plays a huge role in swallowing. It also is involved in our gag reflex. As a matter of fact, testing your gag reflex is a great way of seeing how tonified your vagus nerve is. It also provides parasympathetic supply to the heart, so it can help reduce your heart rate. One of the reasons why we've been paying so much attention to heart rate variability in the last couple of years. It regulates contraction of smooth muscles and glandular secretions in the intestines. And it has receptors in the esophagus, stomach, proximal small intestine, and even has sensory endings in the liver and pancreas. The vagus nerve is responsible for regulating internal organ functions. So we're talking digestion, heart rate, and respiratory rate. It is responsible for vasomotor activity and certain reflex actions like coughing, sneezing, swallowing, and vomiting. Now, I brought up and wanted to tie in the gut-brain axis because I'm sure, as you all know, as functional medicine providers, that there's definitely been an uptick in the presence of functional GI or gastrointestinal disorders, or FGIDs. Now, these disorders are kind of grouped and defined by a unique constellation of symptoms, of chronic GI symptoms that all seem to play a role in these conditions. So the main GI symptoms that tend to be shared across these different disorders are abdominal pain, dysphagia, dyspepsia, diarrhea, constipation, and bloating. What researchers have found is that these conditions arise due to the alterations in the way in which the gut and brain communicate. So for whatever reason, during the development of these conditions, as we know, 90% of the communication comes from the gut and the brain. So for whatever reason, our chatty patty friend, the gut, is not able to communicate with the brain as effectively as it could, and the same can be said for the brain communicating with the gut. Now, these conditions account for over 30% of gastroenterology outpatient visits. They're often associated with chronic pain, like oftentimes patients with these functional GI disorders will also struggle with fibromyalgia, chronic fatigue syndrome, and other functional syndromes. Now, of course, I am sure if you have attended one of my webinars before, you know that I love participation. So we have a pop quiz. Which of the following is considered a cause of gut-brain axis dysfunction? A, LPS endotoxemia, B, fasting, C, candida infections, or D, none of the above. I'll give you all a few moments to put in your answers. All right, we have A and C's. All right, great job, everyone. Great job. The answer is LPS endotoxemia. It is considered one of the primary causes of gut-brain axis dysfunction, and we're going to spend a couple of moments talking about how that actually happens. So lipopolysaccharide is a toxin that's present on the surface of gram-negative bacteria. It's an endotoxin, and because it's present on gram-negative bacteria, that is a huge thing to note because gram-negative bacteria make up over half of the bacteria in our microbiome. So half of the bacteria in our gut have LPS. Now, that's not generally a problem because when it's inside of the bacteria, it does not cause harm to us. But when the bacteria die, they lyse open and they release LPS into the lumen. It can cause inflammation in the gut, and if it seeps out of a leaky gut with weakened tight junctions and is able to enter circulation, one of the very first places that LPS is going to is the brain. And in the brain, it has been known to wreak havoc. In fact, this study found that LPS from gut bacteria caused an inflammatory response that plays a role in the development and pathophysiology of major depressive disorder. Participants with MDD tended to have elevated levels of pro-inflammatory cytokines, poor cellular immunity, and tryptophan depletion. What they found is that systemic lipopolysaccharide exposure increases those same inflammatory cytokines and can cause chronic central neuroinflammation. So think of your brain kind of being on fire, and that is being caused by lipopolysaccharide and the inflammatory process that it induces. In fact, in this study, they looked at microbial mapping of the stool of patients with anxiety and depression, and they found that those patients with anxiety and depression, they had far fewer short-chain fatty acid producers. So they were missing our favorite keystone Faecalibacterium species, but they also saw an increase in bacteria that are associated with inflammation. So they saw way more enterobacteria, desulfovibrio, which if any of you have run our BiomeFX stool test, the common pathogen that falls into this taxa is Bilophilo-Wadsworthia. So if you've seen Bilophilo-Wadsworthia, then you've definitely seen an LPS containing microbe on your stool test. Now, there were some confounding information that is requiring a bit more investigation because further now they have to look at how diet, psychiatric medication, and even gender can also impact some of these patterns that they saw in the microbiome. Now, LPS is not just implicated in mood disorders like anxiety and depression, but it is also implicated in Alzheimer's. This study found that gut-derived LPS has a unique affinity for the nucleus of Alzheimer's disease brain cells. In fact, LPS was found to bind to the nucleus and prevent mRNA communication in the Alzheimer's brain. And it is thought to be one of the main drivers of the development of amyloid beta plaques. And in this study as well, we saw that patients with Parkinson's disease did have a altered gut microbiome and displayed certain intestinal pathologies. What they found is that the inflammatory response associated with leaky gut actually also increases further gut permeability and exposure to endotoxins. So in short, let's do a summary of kind of what happens to the brain when it's exposed to lipopolysaccharide. So as we mentioned here, LPS is a pro-inflammatory endotoxin on the outer membrane of gram-negative bacteria. It can translocate across the leaky gut and activate immune cells. When it's activating those immune cells, it's going to start an inflammatory cascade, which features the production of interferon gamma. This process is going to increase an enzyme called IDO. Now, IDO is responsible for turning tryptophan into kynurenine and some of the other sort of harmful metabolites associated with poor tryptophan metabolism. Now, the presence and increase of IDO and the further metabolism of tryptophan reduces the availability of tryptophan to be used to make other important hormones and neurotransmitters like melatonin and serotonin. So let's look at another schematic to really kind of bring this full fold and kind of color this image for you a bit better. So our diet and our microbiome really impact how tryptophan is used in the body, both the gut and the brain. So when we have a microbiome that is full of inflammation, so we have inflammatory and inflammation-inducing bacteria, and say you also have the same patient that is also under a lot of stress, then we're going to drive tryptophan to be metabolized in the neurotoxic pathway. So that's going to activate that IDO enzyme, and it's going to turn tryptophan into quinolinic acid. Now, this is an issue because quinolinic acid is considered to be neurotoxic. And so patients who are struggling with poor tryptophan metabolism will have a more depressed mood and irritability, reduced resistance to stress, poor sleep, and poor concentration. But if we can clean up the stress and inflammation and reduce LPS presence, then tryptophan is now able to be used to make serotonin for a good, strong, balanced mood, and melatonin for restorative sleep. After cleaning up the gut and improving dysbiosis, we're able to send tryptophan into the neuroprotective pathway, and then our patients are able to have better sleep, a more balanced circadian rhythm, they have a positive outlook, more resilient to stress, and feature a good memory. All right, we're making our way through. Now let's talk about the gut-heart axis. What we know is that dysbiosis plays a critical role in the development of chronic disease, including cardiovascular disease. Now, there have been several studies that have highlighted the crosstalk between nutrition, the microbiome, leaky gut, and the immune responses that affect heart health or promote cardiovascular disease. Now, as I mentioned earlier, the bacteria that is in the gut determines what metabolites are being produced. And so several microbial metabolites have been identified as impacting the development of heart disease, particularly TMA, LPS, certain bile acids, serotonin, and short-chain fatty acids. Some of these metabolites improve heart health, and others can be detrimental. So let's talk about one metabolite that has been pretty common, pretty frequently discussed as of late, which is trimethylamine N oxide, or TMAO. So TMA, as you can see right here at the top of the image, is produced by the gut bacteria from lecithin, choline, and carnitine-rich foods. So if we eat eggs, red meat, full-fat dairy, and fish, then our gut microbiome will take those foods, break them down into choline and carnitine, and then further break them down into TMA. Now, there's really no issue with TMA, but the problem comes when TMA is metabolized into TMAO from the liver or through the liver. And when TMAO is in circulation, it has been found to have many clinical effects, and many of them are just simply not good for our patient's health. So TMAO is a biomarker that is known to be pro-atherogenic. So it is encouraging the development of atherosclerosis, or hardening of the arteries and vessels. TMAO does play a role in atherosclerosis, chronic kidney disease progression as well, heart attacks, metabolic syndrome, and type 2 diabetes. In a meta-analysis of over 19 studies, it showed that higher plasma TMAO levels were linked to an increased risk of cardiovascular events like stroke, MI, or myocardial infarction, and death. What is really interesting is that a higher microbial diversity has been shown to decrease TMAO. So as you are improving the gut microbiome, you're adding more healthy bacteria into the gut, you're able to reduce how much TMAO is produced from that bacterial fermentation. So let's talk about some dysbiosis patterns that we have seen in cardiovascular disease. In 2017, there was a study that analyzed stool samples from 218 people with ACV, or atherosclerosis, or atherosclerotic cardiovascular disease. And they also studied and looked at the stool samples of 187 healthy controls. What they found is that the patients with atherosclerotic cardiovascular disease showed an overabundance of enterobacteria, so E. coli and Klebsiella, and they showed an overabundance of streptococcus. They also saw a depletion of butyrate-producing bacteria like the Keystone fecale bacterium prosonythiae, and they showed a presence of inflammatory metabolites and toxins like TMA, TMAO, hydrogen sulfide, histamine, and LPS. They also saw that patients with atherosclerosis had reduced fermentation of dietary fiber and reduced short-chain fatty acid production. What's really been the most fascinating is that researchers have found that bacterial DNA has been found in atherosclerotic plaques. So it does tie in an implication and connection between our gut microbiome, leaky gut, and the development of cardiovascular disease. So let's take a moment to look at some of the changes that they saw in the microbiota and some of the gut microbiota metabolites that are involved. So as we mentioned with atherosclerosis, they saw an increase in streptococcus, certain ruminococcus, and clostridium. TMAO is most involved in atherosclerosis. And then we saw that some of the mechanisms of TMAO in this process is its activation of inflammation, primarily through activating NF-kappa B. Now we saw some similar patterns with hypertension. So with hypertension, we saw that they had increased Prevotella, increased Bifidobacterium, and increased Lactobacillus. We also saw an increase in short-chain fatty acids, propionate, and even some bile acids. Now, what's interesting is that when they knocked out certain genes, like GPR41, which impacts short chain fatty acid production, that led to an increase in high blood pressure. Certain bacteria, like Bifidobacterium and Lactobacillus, can impact Th17 levels and can lower blood pressure. And then, of course, the bile acids can also regulate the calcium ion concentration, which does also have an impact on blood pressure and heart rate. And then we saw some other patterns here with heart failure. This is slide 30. So once you get these slides, if you want to take another look through this pattern, you will be more than welcome to. So let's talk a little bit more about coronary artery disease. So we talked about atherosclerosis, but what does dysbiosis look like in coronary artery disease? Of course, this 2019 study showed that gut microbial composition and the metabolites made by that bacteria do change significantly with coronary artery disease, and specifically with the severity of coronary artery disease. The gut microbiome of patients with CAD showed that they had an increased abundance of proteobacteria. So remember, proteobacteria are bacteria that contain LPS. So we're thinking about Klebsiella, Streptococcus, and some others. We also saw an increased production of toxic metabolites, particularly TMAO, and a decrease in short-chain fatty acid production, particularly butyrate. We also saw similar patterns in hypertension as well. So hypertension, as we know, is the most common risk factor associated with cardiovascular disease. And the gut microbiome has been shown to play a role in blood pressure regulation. And compared to healthy individuals, those with hypertension tended to have a microbiome that had a low diversity. So we're starting to see a little bit of a pattern here. Increased abundance of the Prevotella gene, or genus, imbalances in the abundance of Bifidobacterium, Lactobacillus, Streptococcus, and E. coli. Now, this is important because, like we said, certain bacteria make different neurotransmitters. And those neurotransmitters can impact our autonomic nervous system, which controls our vascular tone, our heart rate, and subsequently our blood pressure. What we know is that elevated TMAO is positively associated with hypertension, and those with hypertension also showed a reduced production of short-chain fatty acids. What is interesting is there was another study that did a fecal microbiota transplantation from hypertensive patients to germ-free mice. So these were mice that have never been colonized by bacteria. They're completely germ-free. And when they received fecal microbiota from hypertensive patients, their blood pressure also increased, which again draws another line of correlation between the gut dysbiosis and pathogens and toxins present in the gut with heart disease and specifically hypertension. All right, we're making our way through. Let's talk about the lungs. All right, so the lungs are really unique. So microbial dysbiosis within the lungs and gut, particularly in asthma, it can be caused by many factors, including lifestyle and environmental exposures. Now, most studies have underscored the fact that the healthy microbiome in the lungs is very transient and at steady state. But during disease and inflammation, that's when pathogenic bacteria are able to colonize the lungs. It leads to dysbiosis and subsequent release of metabolites that can cause lung issues. So here, this is a unique study that showed the role of the lung and gut microbiota with asthma. And it's interesting that they found that the effects of the gut microbiota on asthma are partially mediated by bacterial metabolites. Again, these are the metabolites that are going to increase our immune system, increase our immune responses, and can lead to an overactive immune system that is quite reactive and responsive to the environment. All right, so the most known metabolite that is known to demonstrate a protective property in the human airway and is able to be protective against airway inflammation is short chain fatty acids. Children with high amounts of butyrate and propionate in their feces at 1 year of age have significantly less atopic sensitization and are less likely to have asthma between 3 and 6 years old. This schematic represents asthma phenotypes and differences in microbiota between these endotypes. So these are kind of the different phenotypes of the ways that asthma can manifest clinically. And then when we look at the different endotypes within asthma, we have type 2 and non-type 2. What they noticed in terms of the microbiota is that patients with type 2 asthma had increased Haemophilus, Moraxella, Klebsiella, and Streptococcus present in their stool. But they also had reduced Streptococcus and even the Actinobacteria phylum, which is where we would find Bifidobacteria. In non-type 2 asthma, they had increased pathogens, including Fusobacterium, which is one of the most inflammatory groups of bacteria. But then they had decreased in certain families, but including the Lactobacilli family as well. So what's interesting is that these endotypes help classify asthma based on certain mechanisms and also have an impact on their disease presentation as well. All right, so that's how the gut microbiome has been implicated in lung health, particularly as it pertains to asthma. So let's talk a little bit about the endocrine system and the connection between the gut microbiome, and particularly as it pertains to diabetes. So the gut microbiome not only helps regulate our hunger and satiety hormones, but it also has an impact on insulin release and insulin sensitivity. Now, there have been several studies that have highlighted the connection between the gut microbiome and glucose dysregulation, and they've noticed that the presence of certain bacteria, certain keystones, like Lactobacillus fermentum and plantarum, Lactobacillus casei, Roseberia, and Akkermansia, have all been shown to improve glucose metabolism. They also improve insulin sensitivity, all while suppressing pro-inflammatory cytokines. So they're able to kind of wake up the metabolic system, get our body to use glucose a bit better, but also cool the fans of inflammation. Now, what's interesting is that metformin, which is the first-line pharmacotherapy for type 2 diabetes, it has been shown to act primarily in the gut and has been shown to alter the composition of the gut microbiota. Metformin has been shown to increase butyrate production. Butyrate is our primary energy source for our colonocytes. It also increases propionate production, which is a very important short-chain fatty acid for our immune system, and also has been shown to increase levels of Akkermansia muciniphila, a very important keystone that helps rebuild our mucosal layer. Now, metformin doesn't just do all of those beneficial things, but it also can lead to certain long-term changes in the microbiome. So at the phylum level, we saw that patients with type 2 diabetes who were taking metformin, they saw these were just patients with type 2 diabetes in comparison to a control. They had a greater abundance of firmicutes. Now, firmicutes and Bacteroidetes are the two major groups of bacteria in our gut, but we like to see a bit of a balance between the two, a 1-to-1 ratio. And patients with type 2 diabetes have been found to have an elevated amount of firmicutes compared to Bacteroidetes. They also had way more proteobacteria than the control group, which means they had more LPS and subsequent intestinal inflammation. They had a reduced abundance of Roseburia. We just talked about Roseburia a couple of slides ago. It's one of our butyrate producers. And then we also saw that the abundance of gram-negative bacteria was also increased with type 2 diabetes. Like many of the other conditions that we talked about before, anxiety, depression, Alzheimer's, cardiovascular disease, and hypertension, type 2 diabetes patients also had low diversity. So low diversity and the presence of pathogens have been a common theme throughout many of these systems in their response to the presence of dysbiosis. Of course, dysbiosis doesn't just affect diabetes and glucose regulation, but it also plays a role in thyroid. So what's interesting is the gut is a target organ for our thyroid hormones. It has receptors for both T4, thyroxine, and triiodothyronine, T3. Thyroid hormone receptors are expressed on the surface of our intestinal epithelial cells, and researchers have found that a disruption in the microbial composition in the gut has been associated with Hashimoto's thyroiditis, Graves' disease, and Graves' orbitopathy. Intestinal alkaline phosphatase is a really interesting enzyme. It's an enzyme that is made in the gut, particularly in the small intestine. It's a brush border enzyme. And it is important because it does play a role, or is tied to thyroid hormone. Now, IAP works by detoxifying, that's not a word, detoxifying LPS. So it really helps our body kind of take down the inflammatory action of LPS and sort of neutralize it. But intestinal alkaline phosphatase in the gene is T3 responsive, which directly links its expression to thyroid hormone levels. So this suggests that if you have a patient who has hypothyroidism and they also have a dysbiotic gut, they may be less likely to make IAP, or intestinal alkaline phosphatase, which means that their body is less capable of clearing LPS and detoxifying LPS. They were able to confirm this in animal models as well. So in animal models, they found that they had lower and decreased levels of IAP gene activity in hypothyroid rats. In fact, when they knocked out IAP in zebrafish, those animals became more susceptible to LPS toxicity. And so if you have a patient with hypothyroidism, it is critically important for you to make sure that you're staying on track with their gut health and clearing out any pathogens that may be present, particularly those that contain and produce LPS. We've also noticed some microbial patterns in autoimmune thyroid disease. Some bacterial strains that are associated with a decreased expression of TPO antibodies and thyroid receptor antibodies. These include Bacteroides, the Dorea species, and our favorite keystone, the Calibacterium prausnitzii. Now, there are certain types of bacteria that are associated with an increased expression of these antibodies. Now, I want to note that this is likely due to molecular mimicry. So our immune system can get a bit confused and mistake these bacteria for these thyroid proteins, and then those antibodies become overproduced. So those bacterial strains include Blausch's species, Lactobacillus species, Allostypes, and Ruminococcus. Now, there are bacterial strains that have been seen in several patients with Hashimoto's thyroiditis. That includes Lachnoclostridium, Bilophila wadsworthia, and Klebsiella pneumoniae. All right, now let's take a moment to talk about how the gut microbiome impacts our immune system. We know that dysbiosis has been associated with many immune-related human diseases, but in some cases, it remains to be established whether dysbiosis is a cause or a consequence of those diseases. So let's consider autoimmunity, for example. Dysbiosis of the gut microbiota has been shown to be related to a lot of different alterations in the immune system. So gut commensal bacteria, they have a lot of roles in our body. They help us establish a regular innate immune system, so our really front line of protection for our immune system. Now, in turn, dysbiosis of the gut microbiota can cause an alteration in the innate immune system and vice versa. And that kind of shows the way that crosstalk between the gut microbiota and the innate immune system kind of set the stage and foundation for autoimmunity, particularly when the gut is dysbiotic. What we see here in the schematic is that microbial colonization in the GI tract in early life does affect how the T cell populations are developed. So there are different types of T cells, T helper cells. So there's Th1, Th2, Th17, or T regulatory cells. And so our microglia, our brain immune cells, help with microglial maturation and astrocyte activation. They also communicate and have crosstalk with our gut immune cells, so our innate immune cells and T cells. And what we found is that the short chain fatty acids that are being produced actually help produce more regulatory T cells that can serve as fire extinguishers to cool off the inflammation that is produced during infections or when the immune system is activated. Now, of course, we can talk about the genitourinary axis as well. Now, usually, I would like to start with just talking about the vaginal microbiome because in many ways, the vaginal microbiome is just as important as the gut microbiome for our female patients. So in many ways, the vaginal microbiome is the opposite of the gut microbiome. In the gut, we want to see a lot of diversity, but in the vagina, we really only want to see one type of bacteria, and that is lactobacillus. A healthy vaginal pH should be pretty acidic, 3.8 to 4.5, and we found that a healthy vaginal microbiome really has 4 dominant lactobacillus species: Lactobacillus crispatus, Gasseri, Jensenii, and Einers. But be very mindful that Lactobacillus Einers can really go either way. It can be associated with a healthy vaginal microbiome or a unhealthy or dysbiotic one. Now, lactobacillus is critical for a healthy vaginal microbiome. It helps keep our pH nice and low, which lowers our pathogen abundance. Lactobacillus species can also make lactate, bacteriocins, and hydrogen peroxide to continue to kill off pathogens and prevent them from setting up shop and creating biofilms. It also inhibits the adherence of pathogens to the vaginal mucosa by making biosurfactants. These surfactants actually compete with attachment sites. So bacteria like Gardnerella or yeast like Candida aren't able to attach to the vaginal mucosa, and thus the host body fluid can then sweep those pathogens away. The vaginal microbiome is impacted by some of the same things that impact the gut microbiome. So age and hormones, sexual habits, medications, use of certain vaginal products, diet, stress, tobacco use, and of course, the gut microbiome also has an impact on the vaginal microbiome. Of course, there is such a thing as gut-vaginal microbiome crosstalk due to the proximity of the vaginal opening and the rectum and anus. So due to that proximity, there may be and usually is translocation of species that are shed in the feces. So due to that close proximity, it is very possible that the bacteria from the colon can lead to vaginal infections and even urinary tract infections as well. So let's talk bacterial patterns and how vaginal health links to the gut microbiome. Women with BV-associated bacteria in their stool, usually Gardnerella vaginalis, Prevotella bivia, or Atopobium, they also show a higher incidence of BV. So these women already have their bacteria in their stool, and due to proximity, it is causing vaginal infections. So when you have a woman, particularly someone with recurrent BV or recurrent vaginal infections, you should also make sure that you are addressing the gut while you are addressing the vaginal microbiome. Now, by inhibiting the growth of unfriendly bacteria like E. coli, you can use oral probiotics to help facilitate and crowd out those pathogens. Women with higher concentrations of Lactobacillus crustatus in their stool were more likely to have a healthy vaginal microbial composition. So let's take a look at how the microbiome impacts the bladder. So there is a bladder axis crosstalk. So with the gut, you will see that they do have certain pathogens like E. coli and Gardnerella in the gut. It is exposed to the bladder. And so you see altered short-chain fatty acid production. The bladder becomes a reservoir for these bacteria to grow. So in a patient with recurrent UTIs, you may see in their stool E. coli, Gardnerella, Pseudomonas, and even some Streptococcus. With interstitial cystitis and BPS, you may see that they have Brevibacterium, Citrobacter, Lactobacillus gasseri, and even Serratia. I'm escaping the UI. I think UI is urinary incontinence. Excuse me, the word escaped me for a moment. But urinary incontinence and overactive bladder has been associated with these species. But interestingly, a healthy bladder is mostly Lactobacillus species. So just like the vagina, the bladder really does not enjoy diversity. It can lead to inflammation and a host of other urinary tract issues. In the urinary microbiome, a healthy bladder has a pH of about 4.6 to 7. It has mostly Lactobacillus and a little bit of Prevotella and some other bacteria. But then if you are exposed to some of these risk factors, so if you have an indwelling catheter, if you're of older age, low estrogen, vaginal infections, existing urinary conditions, and then of course, if you are taking antibiotics, have spinal cord injuries, or even diabetes, that can lead to the development of UTIs. And UTIs, as we saw in the previous slide, have been associated with an overgrowth of E. coli, Enterococcus, and Staphylococcus. But markedly, it is shown to see far less Lactobacillus. So someone who has frequent UTIs could benefit from increasing their Lactobacillus. All right, thank you so much for your time today. We covered so much information and ground. In summary, we have shown that dysbiosis is an imbalance between beneficial and harmful bacteria. Microbial imbalance impacts all areas of our life. And gut dysbiosis has been associated with anxiety, depression, Alzheimer's and Parkinson's, heart disease, asthma, type 2 diabetes, autoimmune disease, vaginal dysbiosis, and urinary tract infections. Ultimately, restoring gut homeostasis is critical for supporting overall health and reducing chronic fatigue. Thank you so much for your attention today. And now let's answer some questions. Let's see. Here's my camera. Hi, everyone. Okay, let's take a look at these questions. First question from Cheryl. What is your take on lorazepam for treatment of leaky gut? I'm actually not too familiar with its use of addressing leaky gut. For me personally, it really starts with identifying who is in the gut. So you have to have a stool test just to see exactly what is in the gut. And then you can model your treatment plan to address that. So it could either include, um, and natural antimicrobials, or you can crowd it out with um, probiotics. And then you definitely want to make sure you're rebuilding the gut. So our total gut restoration kit would be an awesome option for you. So sorry I couldn't comment on lorazepam, but hopefully someone in the comments can share their experience as well. Thanks everyone for your comments. So glad you all enjoyed it. All right, Susan. This is an article that shows associations between MS and Parkinson's disease with a higher amount of Akkermansia in the microbiome specifically. What can you tell us about this? Is it possible that taking Akkermansia poses a health risk? Could it promote the pathology behind MS and Parkinson's? Is anyone doing studies on this? Really great question. Let's start with the first one. There has been, like with any topic, conflicting information with Akkermansia. We do know that it is a mucus degrader, and like anything, balance is important. So we are going to be looking for studies that look at the impact of having too much Akkermansia, whether or not when Akkermansia is overexpressed or overabundant, if it could actually damage the mucosal lining. That is still possible. I haven't been able to dig into the research that much to confirm it, but it's definitely something to think about. When taking Akkermansia orally, one of the challenges is whether or not those bacteria survive the production process. So Akkermansia is a obligate anaerobe, meaning that it cannot survive even in the smallest exposure to oxygen. So even if you're taking Akkermansia, I guess the question remains is whether or not, how many of them are, available and alive and able to colonize the gut, or whether the whole capsule has, dead bacteria in there. So based on what we see right now, Akkermansia seems to be definitely a keystone, something that we want in the microbiome, but we do want balance. So let's just try to aim for supporting a balanced level of Akkermansia. All right. I hope that answered your question. Could it promote the pathology? I mean, it could. If it's eating away at the mucosal lining, it could cause inflammation. And as we talked about earlier today, inflammation and dysbiosis can impact T cells and T helper cells and ultimately the immune system. So it's possible, but I don't know the mechanism of actions or specifics for that. I hope that helped. All right, Mona, what can be done to protect one from the harmful effects of SSRIs or SNRIs, particularly sertraline? Interesting question. I guess it depends on which harmful effects you're speaking on, like whether or not it impacts different neurotransmitter production or anything like that. I wish I had a specific way to answer this question. It's probably best posed to an integrative psychiatrist who specializes in managing SSRIs and SNRIs. For me, when I have patients that are on these drugs, I support their gut by making sure that they're eating good nutrient-dense foods, because we do know that some nutrient deficiencies do coexist with certain mood disorders. And as we saw today, gut dysbiosis does impact mood as well. So even if they're on an SSRI or SNRI, helping in cleaning up their gut can be huge. So I would recommend kind of starting there. Was the 2017 study analyzing stool samples, RCT, and double blind? Hi, BJ. Yes, it was. And when you get the slides, you can get the reference, and you'll be able to read it, and hopefully you'll enjoy it. Okay. Do we know enough yet about these associations, gut organ, to say that microbiome dysbiosis comes first as a cause of organ diseases, or are we still using the association language? If we're saying that it's cause, what would be the top 3 proofs and or classic studies to prove that? Great question, BJ. Honestly, I don't think any researcher can definitively say that gut microbiome dysbiosis is a direct cause. I know that LPS and dysbiosis has been implicated in the development, meaning that it plays a role, whether it's a major or minor role, it does play a role in the development or the different steps that have to take place in order for a disease to develop. So we are still using an association language because we know that correlation doesn't mean causation. So until we have amazing researchers who can directly pinpoint, right now it is just one of the many factors that plays a role in disease development. Hope that answers your question. And Angelica, please let me know if I'm going over time or anything like that. I'll try to get through these as quickly as I can.
Oh, you're okay. We're about 5 minutes over, so maybe a couple more questions.
Cool. Let me look through really quick. Okay, anonymous attendee had 2 similar questions. Is there a probiotic that you make or recommend for the vaginal microbiome? Not currently, but hopefully down the line, we can add that to our repertoire. But usually what I advise is that folks look for bacteria, probiotics that contain those lactobacillus strains. And if you're addressing vaginal health, please, please, please do not put megaspore in your vagina. Only lactobacillus goes there. So those are my 2 recommendations for that. And then your other question was, is there a probiotic that can support eradicating E. coli? What I can say is Bacillus subtilis, which is present in all 3 of our probiotics. It makes its own natural antimicrobials and makes bacteriocins that kill off bacteria. So, E. coli could be one that is susceptible to those same bacteriocins. And then I will answer one last one from Rashmi. What strains can help with histamine intolerance? This is such a great question. If you have caught my Healthy Hormones webinar, I actually have a slide that shows the different types of bacteria that make different neurotransmitters, including histamine. And some of those are actually lactobacillus strains. So that's why it's important that you choose a probiotic that does not have strains that may incidentally be making histamine and might worsen your patients with histamine intolerance. But the strains that can help, megaspore, those spores have been shown to help reduce histamine. So it does not cause or make histamine. So our spores would be a great option to help someone with histamine intolerance, and all 3 of our probiotic products would be a good option. All right, thank you so much, everyone. I hope you enjoyed our time together. And like Angelica mentioned earlier, we have another webinar coming on later this month on BiomeFX. And next month will be our 4th ending to the webinar series for our foundation. So I hope you enjoyed. Thank you so much. And I'll see you all next time.